The overall goal of this procedure is to observe the dynamic assembly of protein complexes in different pH environments using biolayer interferometry and confirm the components using electron microscopy and mass spectrometry.
Identifying and understanding the specificity governing protein complex formation and assembly is of extreme interest to biochemical researchers. The methodology we're talking about today allows researchers to assemble, visualize, and validate predicted macromolecular complexes. The complex nature of the biosensor surface allows researchers to easily release assembled complexes into small microvolumes for electron microscopy and mass spectrometry analysis.
In this demonstration, we follow the kinetics of pH-induced structural rearrangements of the anthrax toxin complex and verify these rearrangements by electron microscopy and mass spectrometry methods, all while avoiding aggregation.
Demonstrating the anthrax toxin assembly on the BLI biosensor surface, macro sample release, and EM negative staining procedures will be Alexandra Machen and Pierce O'Neill, graduate students from my laboratory. Dr. Antonio Artigues is instrumental in analyzing and identifying the BLI released microsamples using the Orbitrap Fusion Lumos mass spectrometer.
To begin, hydrate an amine-reactive second-generation BLI biosensor tip by immersing it in 250 microliters of water and incubate it for 10 minutes. In the Blitz software, program the step times as listed in this table. Start the BLI run by immersing the biosensor tip in 250 microliters of water for 30 seconds to measure an initial baseline of biosensor thickness.